So thanks for introduction. My name is Serghei Glinca. We are Crystals First. What you see here on the title will be hopefully more clear over the course of the talk. So as Frank said, we are so a spin-out of the group from Gerhard Kleber. I've seen there are some colleagues dialed in from our work. So hi. We started the company 5 years ago, and we were initially focused on crystallographic fragment screens, but as a company, we adapted to the market needs. We are now allocated structure biology is in Hamburg at the synchrotron at the DAISY, and we have also a protein production facility, and yeah, a lot of scientific and I mean, we're working now on concepts that have been developed in the group of Gerhard Kleber, and it's they've done a lot of also crystallography work. And I just like to point out the statistically there's 1% roughly deposited by his group. And what is an interesting fact that in the German-speaking region, the Gerhard's book is called The Red Bible of Drug Design. And from what I know, there will be further updates of his book. So what is Crystals First is about? It's like a motto. Crystals First. We kind of advocate. So we are here in the community that we should get structural data and fragments as early as possible. So we're focused on accessing structural data and then apply large-scale computational modeling. Pretty much what Steph was talking about. But when you don't have an optimized soaking system, it might be tedious to get there. So we have a proprietary technology called Smart Soak that accelerates kind of the access to structural data by stabilizing protein crystals to their native state. So it's not cross-linking, and it has been applied to over 40 targets. So it's target agnostic so far, only soluble proteins. And crystallographic, depending on the nature of the protein, crystallographic fragment screens can deliver high hit rates, and those are some logos I can show. So I don't think AViDD needs an introduction. Just a recap. I think Steph has done a great job explaining why AViDD makes a lot of sense in drug discovery. I'd rather focus, use the time to, from what I hear in the industry. So we work with pharma and biotech, and we discuss with medicinal chemists, and there are some opinions why AViDD is not a good option. So, and the goal is to have over the course of the talk to show why it's a good idea to run AViDD. So why not to use AViDD is that there are more efficient methods, right? Like DEL screens, DNA-encoded libraries that are quite fast and cheap and deliver potent compounds. This is the second point. The potency is too low, and which kind of triggers, okay, we need to run chemistry in order to build more potent compounds, going from milli or micromolar to like low micro and nanomolar compounds, which are like HDS-like, lead-like in terms of size. With fragments, of course, we need structures. So in order to build structure-activity relationships, we need structural data. Otherwise, we don't know how it binds and where it binds, and it's kind of a blinded optimization. And structure biology is not always fast, so there are some barriers that chemistry progresses, and then structural data comes in, and then it's kind of helpful to rationalize why it's working, but it's not helping to drive the campaigns. What we also often discussed is that you don't want empty structures, like you saw compounds and the structures are empty, and it's not driving the project, you know, when molecules are not bound to the protein. And that, as I mentioned, crystallography is a bottleneck in general. It has many reasons, but those are opinions. Why not to do AViDD? And hopefully so number one priority at Crystals First to overcome those barriers. A typical fragment screening cascade, I mean, we are here in a community where we screen on crystals, structural data is driving it, but generally it's perceived as a quite expensive screening. And the typical screening cascade has biophysical techniques. So any primary screening, it could be a thermal shift or something else, depending on the philosophy of the group or of the company. And mostly those primary hits are validated by a secondary screen. And then structural biology starts. Compounds are optimized. Computational modeling is working. So it's kind of not the step number one, but step number 2 or 3. With this screening cascade, there are generally problems, and the problem is the overlap of hits. So if you run 2 screens and then look at the overlapping space of hits, it's very low, generally very low. And I'm kind of showing some publications you can use in your presentations too, just to showcase that the question, what is a fragment hit, if you run 2 biophysical screens, might be very difficult to answer if the overlap is low. So those are different techniques, and the mechanisms are different of measurement and readout. So it's a melting temperature or something else. And even for like assay setups, for functional assays, it's also low. And when we were, before we started Crystals First, there was a screening at Gerhard Klever's lab, and Johannes has been driving this work. So they applied 6 different biophysical assays and compared to crystallographic fragments, and they used a established like model protein with a large active site. And also here the overlap was low, even though the site was large as an aspartyl protease. And the bottom line is that a combination of 2 screens as a cascade would have retrieved maximum of 19 crystal structures, let's say, if you would soak only those hits, compared to 71 crystallographic hits. And what is interesting that the combination of all 6 methods leads to 0 overlap. So it's not an easy question to answer, what is a hit, using this approach. And also the idea that crystallographic screen leads only to low potency binders, it was not true in this case. So why put structural data first? I mean, Steph answered it. It captures the potency. It captures the largest spectrum of potency, and we get a 3D structure for structure-based structure design. And to overcome those opinions, again, so why should we do it? There are examples also from companies that have applied a similar approach. So Chimera, they had a millimolar binder, but they had a structure, and they optimized the compounds very quickly just by using a structure-based structure design, not towards just only a potent binder, but also to a degrader for STAT3 and IRAK4. And I believe that STAT3 is quite has progressed further. Haystacks has done a lot of work, but it's interesting that the screening for covalent fragments has been also very successful, and we do a lot of work in the covalent space too. This is my favorite example, and it goes like Boehringer published it in 2020. We started Crystals First in 2019, and they argue for a very similar philosophy. Let's say X-ray first. Again, go even a bit further by saying that medicinal chemistry is biased by affinity. So what I mean by X-ray first is that once you have a new compound, get a structure first, and then look at the potency. And they applied it on challenging target on RAS proteins. And again, they started with a millimolar compound and optimized towards an nanomolar compound. And I think PI 2852 is now a tool compound on their platform OpenMe. I don't think I need to introduce crystallography. I think the for those who are not familiar with that, just to get everyone on the same page, you get co-structures by co-crystallizing or soaking. Co-crystallization without automation is not scalable, and also any change of the crystallization protocol by adding a different ligand might lead to like that the protein is not crystallizing anymore. Our experience was that if the potency is low, you'll get a lot of empty structures. So we prefer soaking. We have some unique capabilities, but the rest of the process is same to get co-structures. A quick introduction of what SmartSoak is. So if you have a reproducible crystallization protocol leading to 1.3, 1.5 angstrom is great, but changing that and getting a soaking system, the standard is actually trial and error, and we have luckily the technology that enables us to set up a soaking system for protocols where no soaking systems are known within 24 hours instead of like weeks or months of optimization. And why it is important is that at low concentrations, so the readout of such an experiment is not just a partial occupancy and we have the signal. Optimally, the system is oversaturated, and at every side there is a binding event. Here's an example of two fragments bound to the same site. At low concentrations, this looks something like noise, and at high concentration, we resolve both binding modes. So we overcome solubility of fragments. We soak at high concentrations. It's a standard setup. It's not always possible to get to those concentrations due to solubility of ligands. So, but we can go even higher, and we strive to soak for longer periods of time. It's not always required for non-covalent binders, but soaking time is, in our experience, critical for covalent. So, as the previous talk was quite impressive. That once you have the structure, what do you do with it? And we have worked with Biosulfite, ChemSpace, Enamine, and Philipps University of Marburg on a kinase, and we were happy that Derek picked up this work and posted on his blog. Was unexpected. So how do we select compounds for fragment evolution? We defined what we call crystallographic ligand confidence. So starting with this snapshot of electron density, the fragment is disordered. So we can place it, but still this phenyl ring is not like it shows still disorder in the electron density. In the center, of course, we have very high resolution, but still not every atom is well resolved, and we wouldn't pick this fragment also. And the one that we picked as an example, the electron density is very well defined. It matches the shape of the molecule, and their occupancy is high. So the definition, so for prioritization for fragment evolution using this structure first approach, is not ligand efficiency or KD. It's high occupancy, the electron density is well defined. It fits the molecule, the electron density reflects the molecular expected molecular shape, and we don't see this order. Of course, pharmacophoric features can be defined, so we wouldn't do fragment evolution for a molecule that has probably one hydrogen bond. It's probably too shaky. So from around 20 structures that we looked for a protein kinase A, we selected only 4 based on molecular diversity interaction pattern. And what we've done, so we worked with BioSelf IT. They have the capabilities to enumerate chemical spaces quite effectively. And you see on the top those 4 structures that we have selected. And then the idea is to use it as a template to dock building blocks with similar pharmacophoric features. So that are filtered after that. So we use, in simple terms, we use the fragments as a template for template docking and filter according to pharmacophoric features. And then the library is enumerated based on those stocked fragments inside the pocket. In this work, we haven't considered flexibility of the protein, so it was rigid protein, rigid docking. I would advise to use flexibility in general, and maybe generate some either post-filtering or pre to consider different conformation, or if there's something known in the literature, to consider that. Then scoring, filtering, clustering is kind of a very common procedure in dot-shell screening. And we synthesized, or not we, like NME synthesized 93 compounds. The success rate was, again, very good. It was around 80%, even higher. And we had tested the synthesized compounds only then in a functional assay, not by a physical assay, but in a functional assay for inhibition. So the space that was enumerated is high, and we use real space. And for the input, we haven't considered any affinity data. First of all, we didn't have it. And second of all, we checked for the inhibition of those initial fragments when we had those synthesized compounds. And the shape that you have seen at the title is kind of derived from this process. It is a quite fast, when you use building block chemistry, it was a quite fast approach. And but the structural biology, like crystallography, has to be established and controlled rigorously. What we have seen after measuring the inhibition is that one cluster was not progressing in terms of affinity, but other 3 were progressing in terms of affinity. And we co-crystallized most potent compounds. So the packing for larger compounds was unsoakable, so we co-crystallized them, like the most active ones, not all of them, and have obtained also the binding modes. The interesting part is that we also compared the functional assay and thermal shift. There are 2 learnings from that. The first one is, in a typical thermal shift screening, those fragments would not provide any signal, no significant thermal shift, like in terms of melting temperature. So those, let's say, inactive fragments, a thermal shift assay would not pick up. And the second is also regarding thermal shift, and there are some conversations with a pharma company. They indicated that they observed that even though your compounds are inhibiting the protein in a functional assay, thermal shift is not picking this up as a signal. And we looked in the data. It's also there were examples where we had as like micromolar inhibition, but no thermal shift. So the learning is thermal shift is cheap, fast, but it's probably a better idea to use thermal shift for larger, like maybe not fragments. Better to use probably lead-like compounds. All right, and as a modeler, for me, it's very important to look at predicted binding mode, and also what is the binding mode in crystal structures, and I pay attention to it. So there are also discussions and questions: do we see a flip, etc.? We haven't seen that, and I think it's mainly due to the selection process that we selected fragments with highest occupancy, and we defined also the interactions, like there were 2 hydrogen bonds, of course, to the hinge region. So the binding mode was maintained compared to initial fragments. Oh yeah, for cluster 4. All right. So overall, we started just only with crystallographic fragment hits. We selected fragments for this chemical space docking based on ligand confidence, not affinity. And you can think of it as a magnet for the needle. So not screening larger haystacks, but selecting the right data to attract similar compounds based on interactions and the chemotype. It was quite successful in terms of hit rate. And I think on a high level, computational tools are very good at calculating enthalpy contributions, but they're not very good at considering entropy. So how we think about it is that with a solved binding mode, the nature solved the most challenging question, the question of entropy. And with high occupancy and less disorder of the binding mode, we kind of use the solution by nature of entropy. And then we use computational tools that are very good at calculating enthalpy contributions. And we went from, you would say, like millimolar, like let's say it's a dead compound, inactive fragment, and no other screen than a crystallographic fragment screen would have picked up this as a hit. And we went from millimolar, so those four fragments that we started with had 5 to 10 millimolar inhibition, which is kind of a noise in a functional assay. And by using this approach in chemical space docking in the selection process of fragments, we had micromolar inhibition, and even one compound was in nanomolar. So a quite significant increase of potency within one iteration cycle. So I would like to comment on the industry's challenge in general. So we are increasing the haystacks computationally, also physically using DNA encoded library, etc. But I think the drug discovery industry is facing several challenges, and we need new approaches, like which was presented in the previous talk, where we get a flexibility in terms of how we manage data and navigate the data that we get. I think the targets are becoming more and more difficult. There's a range of screening technologies from DELS, AViDD, mass spec, HTS. We have a rise of machine learning applications, and projects have to be structurally enabled. There is also a constant conversation: is it a covalent or non-covalent project? Because covalency is not perceived as a threat anymore. It's actually used for design. And of course, we have a rise of heterobifunctional molecules and molecular glues, where structural data is critical. So, and our hypothesis is that the typical screening cascade, which you see on the right-hand side, so you screen, you have some hits, and then we do something with those hits. The assumption is that we always get hits. I think this way of screening cannot adapt to the complexity of drug discovery in general. That's why with the final slide, I would like to what we are building and what is already kind of working in projects that we, use our structure first approach or crystallographic fragment hits. They don't necessarily have to come from crystallographic screening. We can soak even hits from other techniques. And we have developed some pipeline to process data, which is different compared to existing ones. And we're developing chemoinformatics and machine learning models to optimize those compounds, to use the structural data as effective as possible, and synthesize what really makes sense to test in a functional assay. We believe that when compounds progress and become larger, potency is critical, but initially with the primary hits, structural data is critical. And everything that we do, we combine also with complementary approaches. This is what I meant to navigate chemical space. I think this will be the key in future to navigate chemical spaces across different screening techniques, and we will need those tools. And I hope we'll see more of those published in the future. So thank you for your attention. I'm happy to answer any questions.